In continuation with the tapetal membrane, in the secretory type of tapetum, in case of Nigella damascenia, at the tetrad stage, the inner tangential wall of the tapetum disintegrates and a new membrane called tapetal membrane is formed around the tapetal protoplast along the thecal face. It was reported by Bhandari and Ramkishore in 1971. On this membrane are studied the Ubis granule. In asterism, where the tapetum is of plasmodial type, an extratapetal membrane is organized adjacent to the middle layer. Unlike the other cell membrane, the tapetal and the extratapetal membrane are acidolysis resistant. These membranes enclose the developing pollen grain in an anther locule-like sac. The intact pollen sac can be easily isolated by macerating the anther, preferably at the uninucleated pollen stage. It can be done with the help of take few anther in a test tube, add to it a mixture of acetic anhydride 9 part and concentrated sulfuric acid 1 part and heat the tube. During this treatment, the tissue outside the tapetal membrane will disintegrate, releasing the pollen sac. Anther of Cryptosporidium, hibiscus and ocimum require heating for only a fraction of a minute to release the intact pollen sac, but the optimum duration of heating may vary with the plant. Overheating would rupture the membrane. After adequate heating, cool the mixture and add some water. Centrifuge it at a slow speed and discard the supernatant. Wash the sediment with distilled water twice and stain the pollen sac with 0.005% aqueous aniline blue for about 24 hours and mount them in a glycerine. Banerjee 1967 reported that in grass, the tapetal membrane consists of 3 layers. Immediately outside the tapetal protoplast is the fenestrated layer followed by a reticulate layer interconnecting the Ubis granule. The Ubis granules themselves constitute the third layer. However, in sorghum, Christensen in 1972 and Nigella Bhandari and Ramkishore 1973, the membrane does not show the reticulate layer and comprises only 2 layers. According to Bhandari and Ramkishore in Nigella, the layer comparable to the fenestric layer does not stain with the spirit blue, whereas the layer of Ubis body take up the stain. On the basis of the selective staining of one of the layer, the author suggested that the fenestric layer, like the Ubis granule, consists of sporopollenin, but presumably it is a different macromolecular form. The tapetal membrane originates from the secretion of the tapetal cell, which are the most active when maximum deposition of the exene takes place. Occasionally, a direct connection has been observed between the sporopollenin strand protruding from the tapetal membrane and the tip of the spicules of exene. The functional significance of the tapetal membrane orbiculaire wall is not clear. But according to Haslop Harrison 1969, the extratapetal membrane probably acts as a culture sac enclosing the developing microspore together with a very liable tapetal periplasmodium. Chemically and functionally, the tapetal membrane and the extratapetal membrane appear to be similar. Christensen et al. In 1972 have suggested that the orbiculaire wall could be regarded as a vestigial capacity of tapetum is in that phylogenetically this layer was once active sporogenous tissue was described by Bhandari and Ramkishore. Thank you.